Hello, I'm Anna Pumo, and I am now doing my experiment video, still for Lab 2, Aseptic Techniques in Culturing Microbes for Biology 225. I have my E. coli yeast in the epidermidis broth test tubes that have been incubating for about 48 hours, so since 2 days ago. So they've been incubating. I am now going to be transferring some of that broth to the agar plates, each labeled corresponding with the broth tube. So what each broth tube has an agar plate. So what we'll be doing is transferring the broth to the agar plate in a very specific pattern, with the wanting to isolate the colonies eventually. So I will go ahead and start doing that, all being done aseptically. So I'm going to start my little tea light. That is done. I have my alcohol. I did wipe down my counter with 10% bleach, and then I just put some towels over to protect it. I have my 100% bleach ready for afterwards, and of course 10% bleach is going to hang out just in case there's a spill of some sort. So my 3 loops are hanging out in the alcohol here, so they will be used aseptically through this entire experiment. So everything is labeled appropriately. I'm going to go ahead and turn that. Well, we're going to leave it closed for a minute. The first thing I'm going to do is begin to sterilize the edges of the E. coli test tube. Moving that around a little bit. So just get those edges sterilized. Lay that down upside down. So I'm going to go ahead and get the alcohol off of that loop. And each specimen will have its own alcohol, its own loop. So we'll go once in, try not to touch the edges. And I'm going to go ahead and just smear that. I'll take the top off. Take the top off the agar. Just smear this on the top of the agar. I can't really see it. It's kind of a zigzag motion. Put that top back on, back into the alcohol. Again, submerging it into, well, we're going to burn this lip again. Then I'm going to take some of the broth and do a little bit more there. Zigzagging in another quadrant. one more time. Fourth quadrant. My first quadrant's at the top of where my label was. We'll see how that goes. This is going to now go into the bleach. We're throwing that away. The first plate is complete. I'm going to cover that. I look one more time on this. I'm going to be saving the broth specimens for later. I think, yeah, I'm saving the broth specimens. I'm also going to save the agar specimens, I believe. Now I'm going to go ahead and move on to the yeast specimen. So just burning the edges to sterilizing the edges of the yeast test tube, removing one of the loops. I'm going in now to my yeast test tube, yeast specimen broth, and I'm going to do a little zigzag motion at the top of my agar plate, top quadrant. Sterilizing the edges of the test tube, also sterilizing the loop again in the alcohol. So I'm going on to quadrant 2, so I'm going to go back in to my yeast broth and make a little zigzag. Hopefully that is enough. Goes back in the alcohol. Sterilize the lip of the tube again. Let that dry. Back in to the yeast broth. Zigzag motion in the third quadrant. Back into the alcohol. Lip, sterilize lip of the tube again. Let that alcohol dry. Looks like I'm getting some burn, some ash. Well, we'll see what that looks like. And I'm doing my fourth quadrant zigzag on this yeast agar plate. That is actually going into the yeast. It's done. One more time on this lip. Now that could be contaminated with, I don't see it in there now, but, well, I see it a little bit. Okay, that is done with, we're done with that one. I'm going to put the cover on. And these, let them sit for a minute like that. We'll see if I have to, which way they have to be stored. Now to my last test tube, the S. epidermidis. Okay, I'm gonna let that, my last loop. And I'm sterilizing my test tube edges, a tip. Go in, get some of that. I'm gonna start at the top of the agar, meaning the way my label is, that's a zigzag into the alcohol. That's it. Sterilize that lip of the tube again. Can't remember if I did this one or not, so I'm going to do that again. I'm just going to say that's 2. Shoot, I think that was 3. I can't see what I'm doing. Okay, let that dry alcohol. And I did the lip of my tube. Go back in, get some of that specimen, zigzag it in the agar, back into the alcohol. Do this lip of the tube one more time, sterilizing it. Then one more time, one more zigzag. We'll see how the colonies show up in there. This is going to go into the bleach. That is all done. I'm going to sterilize this lip one more time, put my cap back on, and these are going to go into the fridge, these broths, so that they can be saved for another time. I think it's time for the fridge for those. I'm going to go ahead and put this top on. And that concludes part 2 of this experiment video. Those agar plates need to incubate for 48 hours. Another 48 hours is Monday afternoon. I will check them on Wednesday afternoon, which at that time, I should be able to do a results video for lab 2. Okay, that's all. Thank you.